Good afternoon, good morning, wherever you are right now. I'm happy to share you the progress that basically led me to this presentation. I remember like almost 3 years ago, I made another presentation for in under a branch of Australasia, also about Rafflesia. That was actually my actual starting point where I learned about Rafflesia. Now, in this presentation, I will be more than happy to share with you all the progress that I have for these 3 years, or to be exact, almost 8 years while studying with this Rafflesia. Not many people have studied about this amazing flower. Well, I can understand that because the flower itself smells rich. But aside of that, this flower for me is closer to alien than something else for me. So I'm dare to say that this is a really odd plant. But without further ado, let's go. So Rafflesia itself is a Rafflesiaceae itself is a family consists of three genera spread from the southern area of China down to Southeast Asia and including Philippines. It is an endophytic parasite, so you won't be able to see them unless it is the time for them for blooming, for anthesis. Inside the host, when it's still on a vegetative state, you won't be able to see anything until it managed to like burst out and then it blooms. Many mysteries still surround these plants, and we and other scientists are still in progress to understand it more. Of all the three genera, Rafflesia is simply the largest individual simple flower in Pantai, as Alex mentioned before. They are exclusively parasitized tetrastigma, which belong to the grape family or Phytaceae, and the other species, the other genera of Saprophyta and Rhizanthus, they are much smaller with their own characteristic, and each of them only have like 4 species each. While on the other hand, Rafflesia have 41 species up to date in 2023. I took this picture from Plant of the World, created by Kew Garden, and yeah, specifically for Rhizanthus, I already sent a notification to them on behalf of Dr. Hans van Ziger to update the map because Rhizanthus is only found in the Southeast Asia and the western area of Indonesia. So I will talk a bit about the evolution of Rafflesiaceae. The evolution start as the ancestor of Rafflesiaceae emerged. Evolved into Saprophyta, which we suspect it is originated from the southern area of China, and it moved to the Thailand and Myanmar region. The species was originally found only 3, Rafflesia ram as the eldest in the evolutionary line, and Saprophyta poiline, and then Saprophyta Himalayana, which is the largest one, and then the recently found Saprophyta myanmarensis, originated from Myanmar. However, Saprophyta myanmarensis is the only one of the species of Saprophyta that is not yet calculated phylogenetically. And then on the evolutionary line is Rhizanthus. We have only 4 species. The first group belongs to the Deceptor and Lowii. Rhizanthus lowii is the type species of Rhizanthus, and then there's Rhizanthus lowii and Rhizanthus deceptor, and then the second group belongs to Rhizanthus infanticida and Rhizanthus zipeli. And sad news from us in Indonesia, the whereabouts of Rhizanthus zipeli is unknown. Especially because the region where it found originally has been used now for urban development, and we're looking forward to see them again in the nature. Hopefully, they are still there. Rafflesia, on the other hand, is the largest species, has the largest species of 41. One found in Thailand, Rafflesiaceae, some in Malaysia, most of them in Indonesia, and then Rafflesia in Brunei Darussalam, and the other rest, which also consists the smallest species of Rafflesia, Rafflesia consueloae, belongs to the Philippines. This is the glimpse of Rafflesiaceae, if you want to see. Rafflesiaceae arnoldii is the biggest one of all, spanning like 60 to 70 centimeter in diameter. And uniquely, Rafflesia in terms of the what is it? The habitus, I'm not sure. The growth type. Only Rafflesia possess the aerial form, while the rest of them, Rhizanthus and Sapria, tend to be terrestrial. What I mean by aerial form is, as you see in Rafflesia arnoldii picture here, it grows, it blooms in the erect vine of the terastigma. Well, we see Rafflesia arnoldii is the biggest one, Rafflesia consueloae is the smallest one. Rhizanthus, on the other hand, is the weird member of the Rafflesiaceae. It has fur, it mimics the skin of mammal, and it is the only one that excrete extrafloral nectar. It's in here if you want to see. If you consider wait, I'll show you. It's in here. In all of this structure, there's a nectar gland. Sometimes we believe we still have no idea why it is there, and often small insects like ants or bees are tend to attract to there. While on the other hand, the gametic region, where it's like stigma and the anther, are located in here. Yeah, so yeah, it's really odd. Even for me, we're looking forward to study it more soon. And sadly, Rhizanthus is the only genus of Rafflesiaceae that is understudied right now. While on the other hand, in some region in Southeast Asia, Rhizanthus is the only, especially Rhizanthus lowi, it's the only species that is been like overexploited, used as an ethnobotanical medicine. The eldest one, Sapria, this is Sapria Himalayana, and this is the oldest one in the all phylogenetic tree so far is Sapria ram, located in Kaeng Krachan National Conserve in Thailand, and this is in Chiang Mai region. This is just a small glimpse for you, the distribution of Rafflesia. You can see the largest species of Rafflesia are located in both Malaysia, Indonesia, and some in the Philippines. However, the smallest Rafflesia only detected in this northern area of the Philippines. So far, I made a hypothesis about this, saying that there is a possibility that the small flower would be related to the region because, as you can see here, the small Rafflesia is located in northern Philippines, and it matches to the location of where its origin. I mean, in Thailand, there's a Sapria and then Rhizanthus. They only have a small flower, not larger than my palm. So I made this hypothesis: if maybe the cold temperature is affected by winter in Philippines and Thailand is related to affecting influence the size of Rafflesia. But for now, we don't know. As I mentioned before, Tetrastigma is the exclusive host for all Rafflesiaceae. There was once a claim that Sapria also grow in phytis, which the grape, but those claims is unproven. So far, only Tetrastigma, a single genus, is confirmed to be this also host of Rafflesiaceae. This is the odd part about Rafflesia. It was believed by study by Pelser et al. That the early Rafflesiaceae ancestor emerged in Cretaceous era around 100 million years ago. But then the host Tetrastigma emerged only 4 million years ago. So we have a question like what happened in this 60 million years of evolution? Are they not parasitic back then, or they are parasitized something else other than Tetrastigma? This is still a mystery. Even for this, the life cycle of this genera is surrounded in mystery. In Rafflesiaceae and Sapria, we have no idea so far how it developed inside the host. Even the way of their seeds germination is still unclear. What metabolites trigger the germination, for instance, and then what happened inside the host until it crack open from the bark of the vine, the Tetrastigma vine, and then they grow into anthesis. We have no idea. So far, the only thing that we managed to collect for information only for when the Rafflesia is big enough. I say like it's almost swelling in the host. I managed to conduct the histological analysis back then in 2018 and 2019. I will show you later. Before that, we have no idea what happened. This is something that we trace about to learn more about this in order to consider this. As I mentioned before, this is Rhizaanthus. It is honestly, it is still widely sold online. And yeah, I won't tell you where, but yeah, this is something that we still have to raise concern. It is claimed to cure coronary disease, as you can see here, hemorrhage, something, even arthrodesia. When I tried to use in silico analysis, they tend to be true. However, some of the metabolites that belongs to Rafflesia is not really entirely unique so far. And all of this compound basically is you can find this in other plants. Yes, this is something that I also discussed with my professor in Thailand. Rafflesia produces caffeine in nicotine, but this is not something really strange because if I read from based on the literature, nicotine also found in the Equisetum plant, the horsetail fern. So this type of alkaloid is not really exclusive to Solanaceae. In total, there's 21 metabolites, and so far, based on our analysis, only one metabolite here, 1 to 6 triglyceride glucose is unique, but if it's true or not, it's remain dubious because we need further confirmation. Yes, on the in vitro studies, they tend to work as antioxidant, while healing and antibacterial. The genomic features is really strange for me. It has long introns, which indicate like Rafflesia have been adapted in evolution for a long time, and they also streamline their genome. Of course, they like throw away their photosynthetic genes. I have a lot of transposable elements, and the genome of plastids are fragmented, and we still have no idea why is that happened. All that life I will share in this presentation are only the fatty acid metabolism. So why should we like studying this weird parasite? Number one is to understand the evolutionary pattern of plant parasitism itself. Number two, well, we don't have to go up there as our main focus because we're like limited in budget. We haven't go to the other body of space out there. But if you want to learn about some weird biology, you can just go study Rafflesia. That's what I suggest. Number three, there's many things need to be unraveled from the molecular level, medicinal aspect, the metabolite interaction, something like that. And of course, because they are unique, we have to conserve this plant in the wild. In the database of NCBI, so far the available omics data is still limited. The one and only reference genome of Rafflesia belongs to Sapria himalayana, And then it also have like a transcriptome curated by Wu et al. Just last year. Also from a shout out to Professor Wan in Malaysia, who curated the collection of Rafflesiaceae. And then one of my collaborators and my mentor, Jean Molina, who also start to gather the information about Rafflesia species. The rest of them are still limited and require the and deeper sampling for transcriptome and even genome data. This is something that I want to share with all you guys. My works since 2000, let's say the official one from 2018. Although I already started from 2015, but it didn't have some significant aspect. Number one is structural analysis. My focus back then was to understand the histological aspect of the flower. Here, I'm combining the study from me and Dr. Molina. From 2014, a previous study from Nikolov et al. Has initially and inspired me for doing this research. He and his team managed to find what is it like the endophytic form of Rafflesia inside the host. The problem is the view is only limited by transverse cross-sectional analysis. Well, later in 2019, in my finding here, in my discovery here, we try to cut the host along in longitudinal position, so we can see the distribution of the Rafflesia in there. As you can see, compared to the surrounding host tissue, Rafflesia tissue has the very large nucleus, and then we still have no idea how is it like to distribute inside the host. So far, we hypothetically think that it is spread as the surrounding tissue squeeze it out, as Rafflesia is located only in the xylem and cambium era. But it was then disputed by Dr. Ng from Malaysia. He said that the plant tissue is robust enough not to act like an animal cell with squeezing the surrounding. So we're back to square one. We have no idea how is it distributed. One idea is it has a stage where it's only a single cell that able to like grow in a long form, but then the predecessor cell like dying and then it go forward as if it's like walking inside the vasculature of the host. This picture, the second one in the grayscale picture here, this is the transmission electron microscopy photo of the Rafflesia cell. The letter P here is the whatever remain of the plastids of Rafflesia. As you can see, there's no like thylakoid or grana or something that mark the chloroplastic organelle of the organelle itself. It's just like something really odd in there. My other mentor, Ibu Sofi Mursidawati, also tried to grow Rafflesia as in tissue culture, but to no avail. The only successful attempt was by Dr. Lazarus Agus Sukamto back then in 2010. But once the cell reached the callus tissue, back to callus tissue, it goes nowhere. The team tried to induce with auxin or something, but to no avail. And Rafflesia, strangely, it has no phloem. It only has xylem and the surrounding parenchyma tissue. The surface of Rafflesia itself is protected by like papillated cell, as you can see in the E here, and then in G, while on the other side, which protect the entire Rafflesia before it blooming, as you can see in F and in H, it's smooth and surrounded by the flattened cells. All consistent inside this is the perigone, the petal structure, and this one on the right here is the inside area of the Rafflesia, and this is the the base of Rafflesia, the H and I here. And then as the sequencing technology progressed and I managed to get a collaboration with Dr. Molina, I continued to study with omics. I used this and the bioinformatics analysis here. Shout out to my colleague here, Pak Arli Adhityo Wicaksono from I3L, and then my colleague Reza, Teresa, and yeah, this study is also have a sentimental side for me because this is my study, which is my last collaboration with my friend Arfan. He passed away in 2022 before this paper has been published. In the end, from this paper, we managed to find the fact that Rafflesia is in fact possess some some medicinal aspects, including anti-cholesterol, anti-cancer, antivirus, especially for respiratory, and it managed in some degree to be antifungal. However, the metabolites are not that unique. Almost 90% that we use belong to the other plants that we can use instead of Rafflesia while keeping them safe in the nature. The metabolites, 20 of them, of 21, are found from these plants. Berries, where you can find the anthocyanins, coffee, pecan, tea, where we can get the alkaloids and the rest of the phenolic compound, including tomato, gooseberry, and apple, especially Indian gooseberry, where we can find the glucanins. Combine them together, we can say, okay, we can sell some this concoction, artificial concoction that's okay, this is the mimic of Rafflesia. So we can just keep Rafflesia involved without disturbing them. Can use any of them. And then this is one discovery that is also amazed me. That previously we have no idea what is inside the plasmid of the plastid of Rafflesia. But then in this study, we managed to discover that inside the plastid, the only thing that remains is the fatty acid metabolism. So, and then in 2023, Dr. Suilian Guo and her team managed to confirm that discovery. But however, we have no idea why Rafflesia accumulate but basically to keep this plastid containing the fatty acid metabolism only. Of course, it remove the photosynthetic function, but it has no starch function, as you can see in D. This is only Rafflesia. Rafflesia is absence of starch biosynthesis gene. It has no carotenoid biosynthesis gene as well. Only few amount of carbon fixation. But in terms of fatty acid, in the rightmost Rafflesia, especially on the seeds, it has some of the gene strongly active. This is also the paper that has been published recently from us. Actually, this is published just this month. We managed to find that there are many bacteria and fungi that is incorporated and collaborating with Rafflesia to help them to grow. We assume that there's some microbial samples some microbes growing surrounding Rafflesia. As you can see here, T is just a tetrastigma, uninfected, and this is the infected tetrastigma. This is Rafflesia seeds, and this is as a negative control is orobanche. We have a lot of this species in here, especially from the Acetobacteriaceae and then the Bacilliaceae, that we thought it could be like helping a lot in the Rafflesia growth during the parasitic process of the host. And yeah, this is what we found. Some Ribaculaceae that is normally found in mouse gut. This, if it's true, and it then further confirmed the fact that Rafflesia could be spread out throughout by the mouse and rodents, because in previous discovery by Jamili Nais and Dr. Hans Manziger, It turned out that the Rafflesiaceae managed to pass through the digestive tract of the mouse. And then this is also from mammalian plant, Bergella. And then from the other plant-related compound like Micrococcus, it also contains auxin promoter and Pseudomonas. It helps the degradation of cell wall. It may be like helping the degradation of the cell wall in the host. And then it is something that is in progress. Currently, I'm doing my research with Dr. Molina, Dr. Meita, Dr. Arlie, and Professor Wan on this trying to identify the microRNA. We try to understand how the gene regulation related to this microRNA is involved during the flower development. Currently, we found some of the genes, including this is the one of the conserved microRNA in plants, mirror 166, and then mirror 171, and mirror 159. These are the most conserved and deeply conserved microRNA in plants. But so far, we try to understand, we try to finish our project on this. And then this is something that we're going to do next, maybe in February. We already got the data already. We want to understand the nature of gene expression of Sapria and the surrounding host. What is the gene activity during the flowering and the parasitism process? However, we don't have enough genome because only Sapria possesses the reference genome, but not in Tetrastigma. Especially for this Tetrastigma cauliflorum, is still in the unknown region. We maybe in the future we will try to do it with the genome, but so far we try to use the super transcript by merging all the isoform to mimic the genome of the transcript. And then this is something that I talked back then in Nantes in September. So why should we study Rafflesia? What is the significance between Rafflesia and the broom rape in the right here? Sapria is something that we have to conserve, and broom rape is something we have to control. They are pests. Some of these, this is Cuscuta, the dodder, this is the broom rape, and this is the facultative parasitism pterospermum, and this is something that we have to protect. Both of them could help us in understand how to protect them and how to control them in the wild. For instance, Orobanche, this is something that I learned in Nantes. The Orobanche and broom rapes basically require some compound to help them germinate, including alveolactone. They belong to group strigolactone, which is produced by the host and then received by the parasitic receptor D14 and Chi2. This strigolactone has been confirmed to be the germination starter for the broom rape. However, we have no idea how is it something similar to Rafflesia or something else. And then we want to understand in a further study if the microbes really help with this. In the broom rape, we believe that some microbes, or even like we spreading around the strigolactone into the soil after the harvesting of, let's say, this Solanaceae and sunflower, it could trigger suicidal germination, where the seed germinates but it has no host, and then enhance the germination but killing the plants, the parasitic plants. But on the other hand, we want to see if there's like microbes contributing for growth of Rafflesiaceae. So we can see here the comparative from broom rapes and Rafflesia. And then we want to see the next, what next on Rafflesia. So we want to see the biophysics, biomechanics. Biomechanics is special for me because I learned this during my PhD. Rafflesia and most of the parasitic plant has a crevice in the surface of the seeds. We have no idea, like, is it to retain water? to adhere to the surface or something, we have no idea. And then we have to further characterize the host area of the Rafflesia inside the host furthermore to see how the stress that is happening in the host could affect the Rafflesia, or like there like a transfer of metabolites between the host and Rafflesia. Number three, this is something that I really want to see. What happened if the host stress and what happened in the Rafflesia? This is something that would be really hard to see because the vegetative state of Rafflesia inside the host is unknown yet. How the nature of them, yeah. And then is there like a communication between the endophytes inside the host in the quantum level? This is something that we want to see later. And then this is something that I also suggest in Europe, but this is like more utopist for me. But by understanding the nature of the parasitic plant, we might like helping understanding to modify the modification of the carcasses combined, especially because the parasitic plant the parasitic plant seeds are very small, and they collect by harvesting them directly to the soil and like spraying back to the soil. It may cause even bigger problem. We can just do something for this. So lastly, yeah, all of these activities, I'm really thankful to all these people, including Alex last time. From 2015 to 2021, I'm really thankful to Bu Sophie, the one and only person to manage to grow 3 species of Rafflesia in Indonesia and probably back then in the world. From now, I'm expecting to see Jean Molina and Steve to able to grow it in the USBG. More analysis will be done by collaboration in the future. Just this last year, another collaborative review paper published it with help in collaboration from Indonesia, Malaysia, and Philippines, and the UK. We published in the Plant People Planet journal. This article is meant to be informative on how can we conserve and what is the state of the conservation of Rafflesia on each different region in Southeast Asia. And then this is my last visit to Nantes University where I learned about the germination here, the germination of Orobanche, the bloom rate. I had a visiting lecture there in Nantes University, and then a visit to Nantes Botanical Garden. That in the future we plan to collaborate to grow Sapria or Rafflesia in there. For me, having a Rafflesia is not really exclusive. Yes, we have to protect them, of course. It belongs and symbolic to some region in Southeast Asia, but it doesn't mean that in collective we shouldn't like interfere to each other. In fact, we have to collaborate in order to find anything new in this about this planet. This is my visit to Pak Chong in St. Stephen International School in Thailand. I managed to like share my knowledge about Rafflesia to the high school student, and then we followed to like a small basic molecular biology in DNA extraction. Of course, we are doing with some particular plant like longan and onion and banana, and of course not Rafflesia, because I specifically say this to Alex. Please don't use anything like that have a strong metabolite because it will affect the effect of DNA extraction. Yeah, there will be more to come. Again, I really love to protect this plant and to learn more about this. This is amazing quote by late Edward Wilson. Every scrap of biological diversity is priceless, to be learned and cherished, and never to be surrendered without a struggle. This is a quote I found in my friend Jean Molina email, and I really love to put this in to share with you to remind us the importance to protect these basic flowers. This is the photo where I sampled the Rafflesia and Sapria, and all of this attempt would be impossible without all of them. This is Pak Joni Hartono, the one that is not from the local community managed to spread Rafflesia from the seeds. And then this is Masofian. He's contributing a lot with the conservation, with talk, and displaying with a picture, amazing picture of Rafflesia. And he's in charge in the community of Bengkulu where to protect the rare flower, Amorphophallus and Rafflesia. This is in the left corner, bottom left corner. This is Christopher Good. We've been he has been helping with the paper and the editing with the books. This is Majin Molina, my mentor. Usofi, also my mentor. This is my friend. And my amazing girlfriend here, Dia, will also help me with sampling of some Again, thank you very much.